Hey everyone, Dr. Jekyll here, and I'm continuing my deep dive into the pathophysiology of Alzheimer's disease. Now, if you were to ask someone from Sydney or Brisbane, Australia, what's the biggest battle in the world, they might say the State of Origin, which is a rugby league competition associated with this image right here. But actually, a much bigger battle going on globally is the battle between Alzheimer's researchers. The debate between, is it amyloid that's the primary and most important cause of Alzheimer's disease, or is it a different protein called tau and tau pathology? Is that what's causing Alzheimer's disease? So that's the battle I'm going to jump into right now. I'm going to wade into the thick of it and help you guys through it. And so it's called the battle between Team Amyloid and Team Tau. So let's jump into it. So, Alois Alzheimer's, over 100 years ago, he's looking at the brains of these patients that had a particular phenotype of memory decline, then cognitive decline, and then death. And what he sees is these giant protein aggregates here. That's an amyloid-based protein aggregate. It's outside the cell. It's kind of huge, and it gets bigger throughout your life. And they're all throughout your cortex and hippocampus. But he also noticed a different kind of pathology, and this actually occurs inside of neurons, and these are called neurofibrillary tangles, and it's a tangle of a range of proteins from within the cell. It's not outside the cell like amyloid is, it's within the cell. And then the third histological feature Alois Alzheimer's described as inflammation, which I use the most important, but I'll get to that, all right? Team amyloid, team tau, team inflammation is actually the third big team right there. That's Alois Alzheimer's drawing right there, but this is an image that I've taken. These are innate immune cells around an amyloid plaque, and they're acting all inflammatory. And red are the immune cells, and green is the plaque. Okay, so let's talk about these neurofibrillary tangles and team tau. So here we have a neuron, and in blue is the cytoskeleton of the neuron. So the cytoskeleton are proteins that make up the structure. You don't just naturally form into a peculiar shape like that. You need proteins on the inside to poke your membrane and build sort of a lattice, sort of a scaffold from which to force the cell into its peculiar shapes. If you had no cytoskeleton, you would actually form a ball. You can't sort of make these amazing processes without really having your cytoskeleton build up. And so we can actually see some of the beautiful detail of these blue cytoskeleton that make up the processes and body of this neuron. Now, if we zoom in, and to zoom in, we have to abandon light and we have to start using transmission electron microscopy. We can actually see here, this is a real close-up of an axon here. We can see that these proteins that make up the cytoskeleton are very structured and very ordered. They are in no way tangled. They are parallel, long tubes, and they're actually made of tubulin, which is fantastic name. Thank you for naming it the correct name there. Now, if we zoom in on just one of these filaments, it's amazing that we can just kind of keep zooming in. We can see that not only are the filaments here, these microtubules, they're fixed together with these little bridges there, but we can see that they actually form these beautiful structures here made out of tiny little spherical-like proteins that polymerize together into an actual tube. And these are some beautiful images using electron microscopy. So this is roughly what we're looking at here. See this donut shapes all stacked on top of each other to literally make a microtubule. So this is a microtubule, and it's made out of tubulin. Thank you for naming the proteins well. And we can see that they form these beautiful cylinder structures, perfect for making a scaffold for the cell. Now, a cool little fact here is we have these little motor proteins that walk up and down the tubulin to bring stuff to different locations within the neuron, and this actually happens in many cells throughout your body. And this is an animation, a fun animation of one taking a vesicle down. Now, this kind of stuff actually does happen, so it's not too far from the truth, this computer animation. But you can imagine, right, we have rough endoplasmic reticulum in the synapse, generating the proteins that are needed for the synapse. Those synapses are breaking down, so we need more protein. Now, in order to make protein, you need RNA, but RNA is made up by the nucleus, right? And your exons are very long, so you need something to transport all the components of the cytosol in the cell body down to those important synapses. And that's this little guy right here. This is kinesin. Kinesin heads down to the synaptic terminal, and dynin heads back up. And that's all powered by ATP. So we haven't talked about tau yet. So where's tau in all of this cytoskeleton structure that we're talking about? Well, in red we have tubulin, and in green we have tau. And what you can see is tau is punctuated all along these tubulin filaments, and that gives you a little hint about what tau is up to. These are beautiful images, aren't they? So if we look at them, here's our tubulin, and we can see they're wrapped all over in these snake-like structures. And it's not very ordered tau. It's not just going along the tubulin or just going around the tubulin. It can actually go in both directions, and it has these tails flying off, and those are tau. That is tau, and it's wrapping up that microtubule and all the tubulins. Now, the way I like to think about it's a little bit like tau is a little bit like the mortar between the bricks. It really sticks the tubulin together, stabilizes it, and makes sure it doesn't break down or disassemble or depolymerize, right? It's really there to lock it in and support it and glue it together. But it also does something else. So in this experiment, we're actually looking at an axon and we've chopped it this way. So now we're looking at the tubulins this way. So they form little donuts, right? So we're looking down the tubulins, not side on at the tubulins. And we can see if you change the fluid that they're bathed in, which is what they did there, you change the properties of the extracellular fluid. Here they're actually changing the pressure, the osmotic pressure. You can actually create densely packed tubulins or sparsely packed tubulins. And what these researchers actually showed was this process is mediated by tau. So tau is not only holding one tubulin together, it's also dictating the space and organization of the tubulins. Now you can imagine you don't want them all packed together like a crystal if you're going to have the kinesins walking down them. So you do want to generate spaces at certain locations, particularly at the top of the axon, to allow transport up and down, for example. Now, there's a problem with that. If tau is locking those tubulins together, how do we do stuff like this? So these are two neurons, and there's the two processes of a neuron, and they're crawling around the cell culture flask, this is, trying to find a connection. I think it cuts just before they make a connection, but once they get close enough to sense the chemicals being released of each other, they lock in together and they form a great connection. So this is neuroplasticity. This is the ability of the brain to change, and it's critical to forming memories, learning how to move and everything, right? And so we need to be able to move our axons and our dendrites and our processes that come off the axon, our neurites sometimes it's shorthand for. We need to be able to move these in order to make new connections, be plastic, form new memories, and so on and so forth. So we can't just have tau locking in our tubulin, so it's a rigid structure that can never change. We need to get that tau off sometimes. And to do that, we use kinases. So kinases are the enzymes that attach phosphates to proteins. So these kinases are going to come in and they're going to attach phosphates to the tau. And that's going to peel the tau off and allow the depolymerization of the tubulin. Now this allows for plasticity. So if we need our axon neurites to move, we need to depolymerize our tubules. So we need to phosphorylate our tau to peel it off in order for us to reassemble in a different direction. So it will depolymerize and then it will repolymerize in a different direction. And then tau can come along later and re-solidify that new direction that it's gone through. However, this is where neurofibrillary tangles come in. So these are actually predominantly made out of tau. So the thing that Alois Alzheimer's identified over 100 years ago, those little tangles within a neuron, those are predominantly made out of tau, and they're predominantly made out of phosphorylated tau. So the tau peels off after being phosphorylated, and then it can oligomerize, again, a lot like the amyloid, form filaments, a lot like the amyloid, and then tangle, a lot like the amyloid, but this time it's actually inside the neuron, and we end up with a neurofibrillary tangle. Now, we call this phosphorylation, we call it hyperphosphorylation, because it's too phosphorylated. We've got too much tau peeling off the tubulin. So now the concentrations of tau that are free floating go through the roof, and now they start to aggregate and stick together and form these neurofibrillary tangles. So this tangle here is this tangle here at the base of this neuron here. Now, nobody would have guessed that there is an NFT worse than the NFTs, the JPEG NFTs. So paying $100,000 for a JPEG, that's a NFT online, but it turns out there is actually an NFT worse than that, and that is this neurofibrillary tangle. You really don't want these to happen in the brain. And that's because imagine the struggle of trying to do this and trying to do this when your axon is full of neurofibrillary tangles. You've almost got no tau because they're all soaked up in the tangles. And that means all your tubulin is now unstable because it lacks tau. Now, so you can imagine that both of these processes, neuronal plasticity as well as your regular transport of essential organelles and nutrients and large molecules up and down your axons and your vesicles up and down your axons is all disrupted. That neuron is eventually going to lose all of its connections and eventually die. So this is a huge problem to get a neurofibrillary tangle. Now, this is where the amyloid versus tau debate comes in. Now, we can actually visualize amyloid and phosphorylated tau in the brain using radioactive ligands that essentially emit radiation that we can then detect in PET scanners, which is positron emission topography or PET scanners. They can actually detect these radio-labeled ligands. Now, we can design those molecules to bind to certain things. It takes a lot of trial and error, but we've developed molecules that preferentially bind to amyloid plaques, and we've developed molecules that preferentially bind to tau tangles. We make them radioactive and we can inject them in and see where the tau tangles and the amyloid plaques are in your brain. So here we can see amyloid at baseline, so this is before any neuronal atrophy has occurred, and this is phosphorylated tau. Now we can see that they actually have different spatial orientations here. So phospho-tau's all around here, whereas amyloid's kind of all over the cortex in this patient. And then if we map brain atrophy, so that's neuronal death, the shrinking of the brain tissue, we can see that brain atrophy co-localizes massively with the tau phosphorylation rather than the amyloid. So amyloid is not predicting where the neurons are going to die. The tau phosphorylation is predicting where the neurons are going to die. So people argue, hold on a minute, it seems like tau is more important to the Alzheimer's disease than amyloid. And this is where this battle starts to form. Now, indeed, we can do some correlations. Now, I'm going to have to walk you through this graph. So over here, we have cognitive score. It's a mini mental state examination. This is just 30 relatively easy questions that you're expected to answer. It might be something like you get a point for knowing what the date is, or if I hold up a pencil, can you name it's a pencil, and you get a point for that. And it's a 30-point scale. And over here, we've got time in months, so it covers 5 years. And then we've got your PET imaging signal. So in red here, if you have a normal PET imaging, so you've got no amyloid and no tau, this is your expected cognitive decline over those 5 years. Very, very mild. Now, in the light blue, we have people who are amyloid positive all over their brain, but they don't have any phosphorylated tau. So in light blue, we can see if you've got loads of amyloid but no tau, phosphorylated tau, your expected cognitive decline is not a lot. You're looking good. You're looking pretty good. Now in green, we have your amyloid positive and your phosphorylated tau in your temporal lobe. So that's the little thumb on your brain. If that's your brain, that's the temporal lobe. It's right there. That's where your memory centers are within the temporal lobe. And so if you have phosphorylated tau in your temporal lobe and you're amyloid positive all across your brain, you actually have a significant expected cognitive decline over the next 5 years. And if you're amyloid positive across the whole brain and you have phosphorylated tau across the whole brain, this is the whole neocortex, so it's the outer layers of the brain there, you can see you're expected to have a very substantial cognitive decline. So another shorthand way of saying what I've just said is amyloid doesn't really predict your cognitive decline. Tau does. Phosphorylated tau is excellent at predicting your cognitive decline. So you can see where we're getting amyloid, team tau debates going on right here. So what's my take on this? I've got to tell you one other thing. There's one other cool thing about tau. Remember how amyloid was autocatalytic? So if you get a little plug, it's going to oligomerize amyloid and promote the oligomerization of other amyloid. So it's autocatalytic. Tau is like that too. And so tau aggregates actually promote tau aggregates in other neurons. But what's super interesting about this is that the neuron seems to be able to release microvesicles. So if you imagine that's the cell membrane, it could bud off into a vesicle, and in that vesicle is some tau tangles, some phosphorylated tau tangles. That vesicle can then go fuse with a neighboring neuron and put in a phosphorylated tau tangle in the neighboring neuron. That neuron will then have that autocatalytic process of phosphorylated tau sticking to it. So tau can actually propagate and spread throughout the brain. And it's actually super interesting. We see if you get phosphorylated tau in one region of the brain, the next region of the brain that's going to get phosphorylated tau and tau tangles will be the region of the brain that this region of the brain connects to. So if I've got a population of neurons here and their axons connect to this population of neurons, if these guys get phosphorylated tau, then because of the vesicle shedding here, these guys will get phosphorylated tau. So what we can see here is that amyloid has now disappeared from this relationship, right? So the phosphorylated tau can now just go on continuing to cause disease without the amyloid playing any part in it, right? We've now got an independent autocatalytic pathological process that can spread throughout your brain, right? That doesn't need amyloid to make that process occur. Okay, so that's very important. So here's my analogy, right? I think we cannot ignore the genetic evidence that amyloid is the initiating factor of Alzheimer's disease. I go into this debate a little bit more in a previous video, but I see it like amyloid is like the iceberg that the Titanic hits, right? But once amyloid has initiated this pathological process, has started some phosphorylated tau, then that phosphorylated tau process can sort of continue without amyloid. Once the ship has crashed into the iceberg, there's no need for the iceberg anymore. And I consider that the water rushing into the ship is like the tau. Tau, phosphorylation, and tangles actually kills the neurons. And so we absolutely need to stop this. There's no point in destroying, there's very little point in destroying the iceberg once the ship has hit it. We need to patch the holes in the ship. Similarly, I think targeting amyloid very late in the disease is a little bit like destroying, with a drug to get rid of amyloid very late in the disease, is a little bit like destroying the iceberg after you've already hit it. We need to start addressing pathophysiological processes that are relevant and are causing neuronal death at that time. And that doesn't seem to be amyloid. Now, the evidence for this is we've had a lot of amyloid targeting clinical trials. So we've had BACE inhibitors, I should say beta secretase, that's shortened to BACE sometimes. So beta secretase inhibitors, we've had gamma secretase inhibitors, those have failed. We've had antibodies that pull amyloid out of the brain. Those have failed. They've pulled oligomers out of the brain. They've redissolved the fibrils with amyloid, and they've pulled those out of the brain. And all of these drugs have failed. I will say recently there was an FDA approval for an anti-amyloid oligomer drug. So it pulls out the oligomers from the brain, and it caused a very slight improvement in cognitive decline. I mentioned that MMSE score. It was sort of in the region of 0.5 point on the MMSE scale. This drug improved you over the period of 18 months. So that is a minor but maybe important improvement. And if I was to overlay the Titanic analogy on there, is imagine you've got a lot of icebergs and you've got a lot of ships. Getting rid of some of those icebergs will help that process, right? So it'll prevent the amyloid going on and damaging new neurons that might induce phospho tau, even though that process probably isn't that important in the late stage of the game. As we know from that correlational studies, that amyloid load doesn't correlate very well with Alzheimer's disease progression. Okay, so that's my take on amyloid versus tau. Quite controversial. Don't take this as gospel. The debate is still out there, and there's some real bigwigs that would disagree with me, and there would be some real bigwigs that agree with me. So the debate is still going on. But let's have a look at tau pathology, because Is it just Alzheimer's disease? Well, it's certainly involved in Alzheimer's disease. So here we have Alzheimer's disease. It's got amyloid pathology and it's got tau pathology. But there's many other kinds of brain diseases that have tau pathology. So frontal temporal dementia is a different kind of dementia. It affects the frontal region of the brain and the temporal region of the brain predominantly. And it's associated with tau pathology for sure, and it's also associated with TDP. This is a different protein. It phosphorylates, it aggregates, and so we end up with frontal temporal dementia disease has tau pathology as well as TDP43 pathology. Don't remember all these little subunits, that's not important. My point is that all these diseases have very different causes, all have tau pathology. So Huntington's disease, for example, has tau pathology, but it's caused by a mutation that results in a very weird Huntington protein. It's caused by a little repeat in your DNA expanding over and over again. So you end up with hundreds of CAGs in your DNA, which creates a very weird protein, which causes damage, but it also results in tau pathology. Parkinson's disease, you end up with alpha-synuclein protein aggregates. You can see Huntington aggregates as well. Protein aggregates are a big part of dementias. So Parkinson's disease has protein aggregates called alpha-synuclein, and it also has tau pathology. And even chronic head injury, if you just whack your head a few times, quite a few times, you end up with axonal injury, and you also end up with tau pathology. Now, what's linking all these disparate diseases to tau pathology? Well, there's lots of theories out there. Now, obviously, my pet theory, now please just take this as a hypothesis, and it's a small part of the whole literature, but I think inflammation is a huge part of that. So inflammation is massive in Alzheimer's disease, is massive in FTD, massive in Huntington's, massive in Parkinson's, and massive in chronic head injury. And inflammation may be the linking thing between all these diseases and tau pathology. Now, what evidence is for there? There's a lot of evidence for it, but let's just have a look. If you inject IL-1, which is just an inflammatory cytokine, if you inject an inflammatory cytokine in the brain, you end up with all this phosphorylated tau. So this is an immunohistochemistry stain of the brain. The brown corresponds to phosphorylated tau, and you can see that this is way more brown than this. And so this is an IL-1 injection versus a saline injection. Now, if you look for neurofibrillary tangles, you actually get massive neurofibrillary tangles just from injecting an inflammatory cytokine into the brain. It's quite a clean experiment. Whereas you don't get those neurofibrillary tangles in the control group. So we know that inflammation is sufficient to induce tau pathology, and we know that all those diseases induce inflammation. So it's fairly convincing that inflammation may be at least one of the pieces of puzzles that links things like amyloid pathology to tau pathology through inflammation. Now, here's another study. Now, this is even cleaner than the previous study. If you just take neurons in a dish, so now there's no other cells, no microglia, no astrocytes, no blood vessels, no neutrophils, you've just got neurons in a dish, and you put on inflammatory cytokines. They actually repeated this with IL-1, but I wanted to show that there's a range of cytokines there. So if you put on an inflammatory cytokine, you increase phosphorylated tau just through putting an inflammatory cytokine on neurons alone. So again, inflammatory cytokines on neurons alone is sufficient to cause phosphorylated tau. Phosphorylated tau then tangles, and then you end up with neuronal dysfunction and death. And in some respects, it's not that surprising because these cytokine receptors activate a huge range of kinases, a whole cascade of kinases, and these kinases could either directly or indirectly lead to the phosphorylation of tau. And so it's perhaps not unexpected that switching on a million kinases inside a neuron results in phosphorylated tau. Now, what would be the evolutionary reason for this? That's a little bit hard to guess at. You could check out the hypothesis. Again, this is just a hypothesis. The phosphorylating tau is essential for plasticity. And after neuronal injury, you need plasticity. You need to rewire your brain if you've got brain damage. And if you've ever seen a stroke patient, they often become paralyzed on half their body because a whole bunch of neurons have died over here, so now they can't move their hand over here. But sometimes it only lasts 2 to 6 months. And then 6 months later, they can use their hand almost as good as new. And that's neuronal plasticity. The neurons have figured out that we need to rewire in order to control this hand because the neurons that were controlling this hand are now not there. And so inflammation would occur during the tissue damage, and so there might be a signal to the neuron we're going to need to rewire, so we're going to need to phosphorylate some tau. Now that's just a hypothesis, but I think that might be one of the connections between inflammatory cytokines and why we need to phosphorylate our tau, and that can lead to tau tangles. Okay, up next I'm going to cover animal models of Alzheimer's disease. So we're going to explore, and this actually starts to support the amyloid versus tau hypothesis going on there. And so we can start to talk about the animal models of Alzheimer's disease.